Welcome, everyone, to this edition of the Early Career Immunology Seminar Series. I am today's stand-in host, Justin Perry, filling in for Tim O'Sullivan. Before we get started, I would like to point everyone's attention to the series' new website, www.earlycareerimmunology.com. That's earlycareerimmunology.com, as well as our new lineup of exciting speakers coming next fall. Also, as a reminder, all of the past series talks are available for your viewing pleasure on YouTube and can be conveniently accessed through this new website. So the reason I'm filling in today for our usual host, Dr. Timothy O'Sullivan, is because he's our speaker. So Dr. O'Sullivan is an assistant professor of microbiology, immunology, and molecular genetics. He performed his PhD work at the University of California in San Diego, then went on to complete his postdoctoral training in the lab of Joe Sun here at Memorial Sloan Kettering Cancer Center as an American Cancer Society Fellow. He then established his lab at UCLA in 2017, seeking to uncover the cellular and molecular mechanisms underlying protective and pathological functions of the innate immune system. Specifically, his lab studies have focused on defining the signals that promote innate immune cell activation and survival in the context of both pathogen and non-pathogen induced inflammation. I think this is really nicely illustrated by some of their recent studies where they identify novel lineages of human white adipose tissue innate immune cells that initiate adipose tissue inflammation and insulin resistance in mouse models of diet-induced obesity. And I would highly recommend checking those out and reading them. So Tim's talk today is transcriptional and epigenetic regulation of natural killer cells. Welcome, Tim.
Thanks, Justin, for filling in today. I want to thank the ECI community for joining us live. I think one of the best part of these seminars is sort of engaging in a wider sort of dialogue with the greater ECI community and forming new friendships and discussions about immunology in real time. And I want to thank the scientific Twitter sort of community for giving us support for this seminar series, which basically started from a tweet that I sent from my couch and now has gone on into its second season with a brilliant lineup of amazing speakers from across the globe. So thank you for tuning in and for your continued support. Today, I'll be talking about transcriptional and epigenetic regulation of natural killer cells. And here we have highlighted artwork, which was recently featured as cover art that was done by Helena Pinheiro, which also I was able to meet through scientific Twitter on the SciArt community. If you ever need illustrations or just want to support scientific art, have a look at those threads and support a local artist to be able to get some cool artwork made. So what is a natural killer cell and how do we think about this in general? So a lot of people are familiar with cytotoxic lymphocytes, more familiar with these CD8 positive T cells. What cytotoxic lymphocytes are going to do is they're going to recognize target cells and they're going to lyse these through release of perforin, which can poke holes in the target cell at immune synapse, and release of granzyme molecules that can then lead to target cell lysis. So a lot of people think of natural killer cells as perhaps just cytotoxic innate lymphocytes that lack a T cell receptor. But they couldn't be further from the truth. There's a lot of differences in how these cells are regulated differently from CD8 T cells. So in the context of T cell receptor signaling, you have antigens presented on MHC class 1, which can be recognized by a specific TCR. And each TCR is unique for that CD8 T cell. NK cells differ in that they can express a sort of stochastic arrangement of inhibitory or activatory Li49 molecules in mice or KIR molecules in humans that can recognize MHC class 1. And based on the balance of these activating and inhibitory receptors signaling, can either become activated or inhibited. So various activating ligands are present on transform cells like tumors, such how they got their name, natural killer cells, because they were the first lymphocyte to show natural cytotoxicity against tumor cell lines back in the 70s. So other activating ligands like NKG2 ligands can be expressed on stressed cells, such as virally infected cells and also cancerous cell lines, which can activate activating receptors on NK cells, leading to target lysis. Of course, I'm not going to be talking about any of these sort of signaling pathways today, but just sort of a brief introduction to some of the differences when studying NK cells compared to CD8 T cells. In the lab, we've been focusing on viral infection, and we focus on antiviral NK cell responses to cytomegalovirus. And the reason we do this is because there's really good evidence that people clinically presenting with NK cell deficiencies have increased susceptibility to human cytomegalovirus, and mice that are deficient in NK cells dramatically succumb to murine cytomegalovirus. And this model system has been really informative in teaching us about the signals that activate NK cells and sort of the parallels between NK cell activation and CD8 T cell activation. So I'm showing you this figure, which was adapted from one of Joe Sun's annual reviews of Immunology. And in here, they sort of review the three signal hypothesis and its parallels to activation of NK cell biology. In the first signal, you have an activating receptor in B6 mice, which is called lymphocyte. And this recognizes a glycoprotein that's expressed on the cell surface of virally infected cells, M157. This acts as an antigen-specific signal akin to sort of TCR signaling. But the difference here is that the activating receptor lymphocyte is germline encoded, so it's not rearranged like TCR. So NK cells exist in the absence of RAG-mediated recombination. They also have co-stimulation, so DNAM1 is able to bind CD155 and CD112, in addition to receiving cytokine signals such as IL-12, type 1 interferons, IL-18, and IL-33. These three signals act together to induce a transcriptional program that leads to activation of NK cells. And this is a similar sort of story in the human. So in response to HCMV, human NK cells expresses activating receptor NKG2C, which can recognize HCMV associated peptides that are loaded on HLA-E. Really nice work that's been done by Chara Romagnani. They can also receive co-stimulation through CD2 signals, and IL-12 is really important for stimulating human NK cell responses to human cytomegalovirus. So we like to use viral systems to understand the basic molecular mechanisms that activate NK cells, because these innate lymphocytes have sort of co-evolved with cytomegaloviruses in order to mediate protection of the host. So what better system than to study the evolutionary response to a virus to understand how a lymphocyte works? The other reason that we study cytomegalovirus is because we know that in the mouse system, so here for MCMV, this specific ligand receptor interaction, this activating receptor lymphocyte, recognizing virally infected cells that express M157, Joe Sun showed back in 2009 that NK cells that are lymphocyte positive, when adoptively transferred into mice that lack this receptor genetically, can undergo a dramatic burst in proliferation. And subsequent studies have actually done this on the single cell level, showing that each individual NK cell clone can proliferate anywhere from 1,000 to 10,000 fold to create an expanded effector pool. These NK cells are able to expand and clear virally infected cells, but now you have a giant population of expanded effector cells, which you don't want hanging around long-term, lest you actually get some off-target immunopathology. So this giant expanded effector pool undergoes contraction and forms what we call a memory NK cell pool. The reason we call this a memory NK cell pool is because these NK cells, instead of having a half-life of 7 days, like normal naive NK cells, Joe has shown that these memory NK cells can persist up to 6 months to a year in adoptively transferred hosts. When they are recalled with another pathogen that expresses M157, they can mediate antigen-specific recall. And when you re-stimulate them with M157-bearing targets, they actually produce more interferon gamma and can have enhanced cytotoxicity. So they have all of the basic properties of a classical memory cell response. We know that this large expanded effector pool is going to undergo contraction in a BIM-mediated fashion. This is work that has been done by Louis Lanier's group, and BIM is a pro-apoptotic molecule that's going to induce cell-intrinsic apoptosis. But what we don't know is why some of these expanded effector cells are going to persist to memory, and why some of these are going to die. And that was sort of a big question that we had when we were starting the lab. And I really like this model system as a fundamental mechanism of understanding NK cell persistence and how naive NK cells can acquire either a transcriptional or epigenetic program that allows them to persist long-term. This is a really interesting model system to be able to interrogate lymphocyte biology with this dramatic increase in longevity seen in the innate immune system. But I want to come back to this original question. Why do some effector cells die and why do some survive? And we know from the T cell literature that CD8 T cells, there's actually dramatic heterogeneity in the effector pool, and this can actually dictate functional outcomes surviving to the memory pool. So based on work that's been done by Sue Keck, Nick Joshi, also work done by John Weary and Ananda Goldrath, we know that the expanded CD8 effector pool is very heterogeneous, stuff that's been done by fate mapping, cell tracing, and also single cell RNA sequencing. These effector T cell pools consist of, here we're showing short-lived effector cells. These cells are destined to die. They don't live as long, and they don't form memory cells as well, here in blue, but also contain a small proportion of these yellow, what are called memory precursor cells that will go on to form more memory cells. And also a smaller population of what are sort of stem cell-like CD8 T cells that have shown to have a history of less replications and are more quiescent, which can then repopulate an effector pool upon restimulation. So we know from previous work that these sort of stem-like and memory precursor pools are important for the continual survival of these memory CD8s. The key difference with memory NK cells is that instead of forming a stable pool over time, they will undergo continual contraction over time. So there was an open question of whether there actually was a similar effector heterogeneity in expanded NK cells after MCMV infection, and whether they would contain some of these quiescent stem-like or memory precursor populations. So a key question that two graduate students had, Luke Riggin was my first graduate student, is now gone on and graduated last December. And we collaborated with Feng Ma, who's in the Pellegrini lab here at UCLA. And they wanted to just ask a simple question of whether transcriptional heterogeneity existed in this expanded effector pool. And in order to address this question, we use the adoptive transfer system that I alluded to before, where we can take wild-type Li4NH positive NK cells. We can isolate these cells using negative selection columns. We can transfer these cells IV into Li4NH knockout mice and then infect with MCMV. And so this adoptive transfer system is nice because you can track your adoptively transferred NK cells. Only the Li4NH positive adoptively transferred cells will undergo this clonal burst proliferation, and we can track where they are on day 7 after they've undergone clonal proliferation. So we harvested the spleen, sorted these Li4NH positive NK cells, and performed single cell RNA sequencing because that's just what you do today. So when we look at the total pool of the expanded effectors, we saw only 2 populations of cells based on initial clustering. And we thought that these 2 clustering schemes were robust because the key differences between these 2 populations came up with Go terms as response to stress, apoptosis, cell death, or programmed cell death. So we thought we were enriching for a certain cell cluster that seemed more predispositioned to undergoing cell death and one that might be more predisposed to survive. And so to test this, we could actually take day 7 cells that we sequenced, and we can combine them with cells from day 14, which are 7 days into the contraction phase. And we can ask whether the same cell state identified here by single cell RNA-seq could actually persist during the contraction phase to identify which subset of the effector pool might represent a cell state that is persisting and could preclude memory formation. So when we look at this, we put it together, we can actually see that most of the cells from day 7 here in red and day 14 seem to have unique gene expression programs. This has been shown by Jo-sun previously. But if we look at the clustering, it forms 5 main clusters here. But I want to sort of key in here on this cluster 3 here in blue. It seems to be equally shared between cells from day 7 and day 14. And this is a really cool result because it suggested that one cell state is preferentially surviving from an effector stage at day 7 to day 14 during the contraction phase. And this could be a cell type that could give rise to memory cells. Here, cluster 0, these are the majority of cells on day 14. We've shown through a lot of in silico differentiation trajectories that these are sort of a terminal differentiated state of memory NK cells. And it seems that cluster 3 is upstream of those, at least from RNA velocity modeling. But I won't get it too much into that today in sake of time. But what we wanted to go back to is we could create module scores from bulk RNA sequencing that was done by Sue Keck and Ananda Goldrath, and we could superimpose either memory precursor or terminal effector, short-lived effector T cell signatures onto these clusters. And what we found was that this cluster 3 was significantly enriched compared to the other clusters in this memory precursor module score. And this was suggestive to us that there potentially could be a memory precursor NK cell that is preferentially persisting during the contraction phase to form memory NK cells at later time points. Using the terminal effector score from CD8 T cells, we found that cluster 0 was sort of more enriched in its terminal effector score, but the score was up in almost all of the NK cells, but less enriched here in the in cluster 3. So that's fine. A lot of people do single cell RNA-seq, and we can see a lot of things in silico, but it doesn't really matter if I haven't proven to you that this heterogeneity matters and is functional in vivo. So we went back to the data set and we looked at markers that were suggested to be differentially expressed between these two different clusters on day 7 to see if we could identify any heterogeneity within the effector pool functionally by cell surface protein expression. And a lot of these were not necessarily bimodal, but we did find 2 markers, CX3CR1 and Ly6C, that seem to contain nice positive and negative populations. We tested CX3CR1 through co-adoptive transfer experiments, and we didn't find a difference in the survival of these cells. So I'll be talking mostly about experiments with Ly6C. So what did we do? Sorry, I seem to be freezing out a little bit. We took a sort of convoluted approach, but I'll explain it in detail. We took congenically distinct NK cells, so mice from CD45.1 or CD45.2, so with a different antibody, you can actually see which NK cells were derived from which spleen. We mixed these at a 1-to-1 ratio, adoptively transfer them into Ly4NH knockouts and hit them with MCMB. And in our expanded day 7 effector pool, we now have an equal, we can sort out an equal mixture of Ly6C negative cells that are CD45.2 or Ly6C positive cells that are CD45.1. We've also done this on either or congenic and gotten similar results. We mix these at a defined ratio, so around 50-50. We put them back into Ly4NH knockout mice that are naive. We've also done this experiment with Ly4NH knockout mice that have already received NK cells and allowed them to expand to day 7 and then looked later. And we wait for about 12 days after transfer, enrich the entire spleen, and then look at how this ratio changes. So is there a survival benefit of one of these cell types versus the other? And what we find is that while you have a equal mix of both Ly6C negative and Ly6C positive cells after the sort, after the transfer, you see an enrichment for these Ly6C negative effector NK cells. So they seem to preferentially survive the contraction phase. If you look at this as a percentage change from the initial ratio, you can now see about 40% on average more of these Ly6C negative cells in an internally controlled experiment. So the majority of the cells that are now persisting into memory time points are derived from Ly6C negative effector NK cells. And we also looked at this by number, so you could tell that even in an internally controlled experiment, we could still derive increased number of these cells from the spleen. So why is this happening? We looked at a lot of different things. It wasn't due to trafficking. It wasn't due to different cell intrinsic metabolism. It wasn't due to differences in proliferation. So then we went to bulk RNA-seq when you can't figure anything else out. And we were surprised to see that the difference between Ly6C negative effector NK cells and Ly6C positive NK cells on day 7 there were very few differentially expressed genes. But what popped out was BCL2. So it seemed that Ly6C negative cells had a much higher expression of this anti-apoptotic molecule BCL2, which is known to inhibit BIM function, than compared to Ly6C positive cells on day 7. There was also a higher expression of this transcription factor ZBTB33, and Ly6C positive cells had higher expression of CDKN1B, which is also a negative regulator cell cycle progression. We've tested both of these with a CRISPR screen and didn't find any role for either of these molecules. So we have followed up on that, but we didn't see anything here. But it really seems to be the only thing of importance here is BCL2. So there's higher MFI by protein of BCL2 in the Ly6C negative effector NK cells compared to Ly6C positive in the same mouse, but no difference in the expression of the pro-apoptotic molecule BIM. And why does BCL2 matter? Well, it matters because, as I mentioned, this BIM molecule is going to associate with mitochondria, and it can send a pro-death signal. However, its pro-death function is blocked once it's bound by BCL2, which promotes survival. And how does this promote survival? Well, in the setting where you have more BIM molecules compared to BCL2 molecules. Sorry, I seem to be freezing here. There we go. When you have more BIM molecules compared to BCL2 molecules, BIM is able to open up and bind backs and back pores on the outer mitochondrial membrane, leading to leakage of cytochrome C into the cytoplasm, which can then stimulate intrinsic cell death. So previous studies that have been done by Nick Huntington's group in Australia have suggested that even though BCL2 can be important, there's functional redundancy for NK cell homeostasis with this gene called MCL1. And it seems that NK cells rely more so on MCL1 during homeostasis for their survival. So it's sort of an open question of whether BCL2 would actually matter during the contraction phase because NK cells can still express MCL1. And he since followed up this paper also by showing that NK cells that are in cycle, they sort of don't have a dependency on BCL2 for their survival, whereas at steady state, you have more of a sensitivity to BCL2 inhibition. So we didn't know what the effect would be after inflammation and whether during the contraction phase NK cells would require BCL2 or whether MCL1 expression would be sufficient to mediate their survival. So we looked at BCL2 expression levels, and it was surprising to us to actually see that BCL2 levels dropped on day 7 in the bulk adoptively transferred NK cell pool. If you look at the MFI here, it's a dramatic reduction of BCL2. And so this sort of suggested to us that maybe BCL2 levels do matter because of the dramatic reduction that you see in the bulk effector pool on day 7. And maybe these cells are actually sensitized to loss of BCL2 levels, which might lead to their intrinsic apoptosis. And we wanted to test this functionally. So we were able to use a technique that's called BH3 profiling. And this is in collaboration with David Nathanson's lab here at UCLA. And the sort of nuts and bolts behind this technique is that you can use a ABT199 inhibitor, which can inhibit BCL2 activity. If you add this inhibitor and your levels of BCL2 are already low to begin with, then you'll have a higher proportion of BIM activity, which can lead to cytochrome C release into the cytoplasm. And now what you do is you permeabilize the cells so that if there is cytochrome C leakage out in the cytoplasm, now this just exits the cell. And so you can look at that by intracellular staining of cytochrome C. And if you have cells that still retain their cytochrome C, we think of these as viable. And the cells that have now released their cytochrome C, we now think of those cells as pre-apoptotic. So we can see the sensitivity of each of these cell populations in response to this inhibitor, BCL2, to see how sensitive they are to BCL2 inhibition. So on day 0, we thought this was really interesting. If you inhibit BCL2 at a certain concentration, you actually don't get any release of cytochrome C, suggesting, similar to what Nick has found before, that BCL2 levels are sort of redundant, you have high levels of MCL1, can still inhibit BIM activity. You don't have any pre-apoptosis. But on day 7, you can see a stark contrast here that when you inhibit BCL2 levels, you now have a massive increase in the amount of cytochrome C that has been released from the cells. And you can see that quantified here. So this really suggests that effector NK cells heavily rely on BCL2 levels in order to maintain homeostasis. And these Ly6E negative, or we're calling memory precursor NK cells, seem to have higher expression of BCL2, which might mean that they can survive better. But I haven't shown you that BCL2 is actually required for the survival of these cells. So we relied on a technique that was developed in my lab, also by Luke, in which we can electroporate primary innate immune cells with ribonucleoprotein complexes with Cas9. We electroporate the cells, we add in an RMP of a target of interest, and we can rapidly delete any sort of gene that we want. So here, using a BCL2 RMP, we achieved over a 70% knockdown in protein. And you can see that our non-template control against ROSA 26 locus still has high levels of BCL2. We can then mix these NK cells, zap them immediately, and transfer them into Ly4NH knockout mice before infecting with MCMB. We mix them at a 50-50 ratio. And then if we look at the adoptively transferred Ly4NH positive NK cells after infection in these recipients, we actually see the ratio of these two populations of NK cells to then surmise what effect depletion of BCL2 has on memory formation. And we can see here that on day 7, we see a significant drop in the amount of BCL2 deficient NK cells. And there's a dramatic depletion of these BCL2 deficient NK cells going into the contraction and into the memory phase of the response to MCMB, which suggests that BCL2 is absolutely required for NK cell memory formation. So to kind of briefly summarize this part of the talk, we have a model of memory precursor NK cell differentiation where we have 2 different subsets of NK cells on day 7. These Ly6E positive cells that seem to be more terminal effector-like, that are sort of predisposed to cell death through lower levels of BCL2, and these sort of BCL2 relatively higher cells that we're calling memory precursor NK cells that I haven't had time to show you, but they can actually differentiate to sort of these Ly6E positive memory NK cells. This we think is a continuous differentiation, and this is why the numbers will always contract, which is a little bit different than what we see in CD8s. But now sort of this mechanism, even though we saw higher levels of BCL2, it wasn't as satisfying. We wanted to know what the molecular mechanisms were that controlled memory precursor NK cell formation. And we know from Joe Sun's work that NK cells in this system undergo a dramatic epigenetic and transcriptional rewiring. You can see here by RNA-seq, based on this PCA plot, that NK cells that are initially activated on day 2 have a completely different transcriptional profile. This changes up until day 4, a more proliferative time point after MCMB, and then even persists for day 7, day 14, but then comes about back to normal levels in similar naive at a memory time point. What's different here, though, is that the epigenetic profile seems to be fixed and distinct from naive NK cells, and at day 14 seems to be very similar to day 35 time point. So this suggests that the epigenetic modifications might be more important in looking at the differences in the molecular mechanisms that might inform us how memory NK cells persist. So we looked at TAC-seq between Ly6E negative NK cells and Ly6E positive NK cells on day 7 after MCMB infection, and found a lot more differentially accessible regions. We then took this signature and compared it with the differences between memory precursor CD8s and terminal effector CD8s, and found a sort of conserved 114 gene signature of differential accessibility of certain genes that were shared between our sort of memory precursor innate lymphocytes and our memory precursor CD8 cells. And when we looked at the binding motifs for these shared differentially accessible peaks, we found an enrichment for ETS1 and FLY1 binding. Whereas if you look at the terminal effector CD8 T cells and Ly6E positive NK cells, you get enrichment for TBET, RUNX1, and RUNX2. And TBET has been shown in the CD8 T cell literature to induce terminal differentiation of these cells. So we thought we were on the right track. And interestingly enough, FLY1 John Weary has shown has actually been really important for limiting effector formation of CD8s in an unbiased CRISPR screen. But we wanted to know if Fly1 was actually expressed in NK cells. So if you find a binding motif, it doesn't really mean anything. So we looked at the transcriptional profile. We can see that Fly1 was actually repressed early after MCM infection, and then becomes de-repressed between day 4 and day 7. So this is suggesting this is a time point that is usually synonymous with when NK cells are undergoing rapid proliferation between day 3 and day 4. So we wanted to look at what are the signals that might be inducing Fly1. And so Joe, of course, has provided a lot of exciting data sets that we can mine for free. And he stimulated a lot of these NK cells with a lot of pro-inflammatory cytokines and looked at the transcription response by bulk RNA-seq and also ATAC-seq. And we found that while IL-12 and IL-18 actually suppress Fly1 expression, IL-2 and IL-15 almost double the expression of Fly1, which suggested that this gene might be induced by IL-2 and IL-15. And if we look at Western blotting for Fly1, we can see that there's a dose-dependent increase in Fly1 protein expression after stimulation with IL-2 or with IL-15, with the most protein accumulation after stimulation with IL-15. So we went back to some of Joe's data sets as well, and we looked at ATAC-seq in the Fly1 locus after stimulation with IL-2 and IL-15. And you can see that there's sort of 2 peaks that seem to be increase in accessibility. And we overlaid these peaks with STAT5 ChIP-seq, as IL-2 and IL-15 signaling uses STAT5 in order to induce gene expression. And we found a significant enrichment at these 2 peaks for STAT5 binding site. So it suggests that Fly1 is likely induced by STAT5 signaling. In order to test that, we stimulated NK cells with IL-15 in the presence of DMSO or a STAT5 inhibitor. And you can see here that in presence of a STAT5 inhibitor, you have much less Fly1 protein accumulation. And this is quantified here, which suggests that IL-2 and IL-15 induce Fly1 expression downstream of STAT5 signaling. But what does Fly1 do? We knocked it out using our CRISPR-RNP approach. We co-adoptively transferred these cells. And surprisingly, we actually saw that there was way more effector NK cells. And this persisted into the memory phase of the response, that the Fly1 edited cells outcompeted wild-type NK cells. And this wasn't due to trafficking or differential persistence in any other peripheral organ, as we saw this in the lung, liver, spleen, blood, which suggests a sort of general enhanced fitness of these cells. And why do I say enhanced fitness? Because when we look at the ratio of BCL2 and BIM levels, which I've shown are very important in establishing the homeostasis of NK cells, you can see that the Fly1 RNP edited cells have much less levels of BIM, which is pro-apoptotic, whereas they have similar levels of BCL2. And this is earlier on, so this is 3 days after viral infection, which is where we see the biggest difference in the change of Fly1 expression levels. So this pro-survival to pro-apoptotic ratio seems to be much greater in these Fly1 edited early effector cells, which suggests that Fly1 is restricting the homeostasis of these early effector cells so they can't form as many effector lymphocytes, likely as a mechanism of limiting immunopathology, similar to what John Weary has shown in CD8 T cells. So to summarize the first part of the talk, we know that IL-2 and IL-15 through STAT5 signaling can induce Fly1 expression. This increases the level of BIM in early effector cells on day 3. This can limit the effector pool, therefore limiting the amount of memory precursor cells that can form and limiting the amount of memory cells. So in the absence of Fly1, you now have less BIM, more BCL2, which increases the survival, an expanded effector pool, and a much greater increase in the amount of memory precursor cells that form, which then allows for the survival of more memory cells. So this study was recently published. And if you're interested in that, I definitely suggest that you read it. But I'm now going to switch gears to another story that is unpublished that we're really excited about as well. And this has to do with sex differences in immunity, a topic I think that a lot of us think about sometimes when we're writing a grant and controlling for gender, but we maybe don't think a lot in terms of functional outcomes. And in the immune response, we actually know that there are a lot of differences between genders in innate and adaptive immune responses. We know that in B cells, in response to viral infection, they actually have higher levels of antibodies, and we see enhanced proliferation and activation of CD4 T cells. But at homeostasis, there's also increased number of NK cells in males. This is both in mice and humans, which is sort of interesting. If you look at the prevalence of autoimmunity, this is almost threefold higher in females, but they also show an enhanced responsiveness to vaccines, whereas the susceptibility of infections is greater in males to certain viruses. And so we were sort of interested in this because this sort of seems counterintuitive, especially for what I've been telling you about for herpes virus family members like CMV. Why would you have more NK cells but have an increased susceptibility to infection? It doesn't really seem to make sense. So we wanted to really focus on why there might be differences and why the NK cell homeostasis might be sexually dimorphic. Can we explain that mechanistically? So this is work that was done by a talented grad student in Maureen Sue's group. We did this in collaboration. And Mandy wanted to look at if biological gender played a role in NK cell functionality. So we confirmed previous studies that suggested that male mice had higher frequency of NK cells. You can see here that the frequency is significantly increased in male mice compared to females, as well as the number of NK cells is also increased in the spleen of male mice. But previous studies, even though they show a difference in numbers, we also wanted to look at effector function. So we looked at these NK cells and we looked at their response to pro-inflammatory cytokines ex vivo. So we stimulated them with IL-12 and IL-18, which I mentioned previously are cytokines that are really important in mediating antiviral NK cell responses through the production of interferon gamma. And you can see here that we see the opposite phenotype now. When you stimulate female NK cells, they make a much higher percentage of interferon gamma than male. So even though there's more male NK cells, the female NK cells in mice not only make a higher proportion of NK cells, but on a per cell basis, they actually produce more interferon gamma than male NK cells. And so this suggested that perhaps male NK cells are functionally deficient in their response to inflammation. We wanted to confirm this in human samples. So from healthy donors and PBMCs, we enriched NK cells and we stimulated them with IL-12 overnight. We could also see a difference in the percentage of interferon gamma between females and males in enrichment for female human NK cells, much greater than males. And also by MFI, you actually have more interferon gamma produced on a per cell basis. So this mechanism seemed to be conserved between mice and human. And we wanted to understand why. And so when you look at sexual dimorphism, there are two things that might be able to explain that. And from the literature, it suggested that a lot of these differences in NK cell number could be attributed to sex hormone-induced effects. So estrogens, progesterone in females, and androgens in males. So one way that you can look at this and control for sex hormones is you can actually gonadectomize the mice. So you can remove these organs, thereby limiting the amount of sex hormones, and then look at NK cell function after gonadectomy. And we were surprised to see that these differences actually held up in gonadectomized mice. So male mice had a much higher number of NK cells in the spleen than female, and also produced less interferon gamma on a per cell basis after stimulation with pro-inflammatory cytokines, as well as a lower proportion of interferon gamma positive cells. So it seems like, at least from this experiment, there are other experiments that we're doing to confirm this phenotype. It seems that the differences that we're seeing in NK cell homeostasis and effector function are not due to sex hormones. So what could it be? We also know that there's a difference between males and females in sex chromosomes. So females have 2 copies of X chromosome, and males have an X and a Y chromosome. Now, females normally undergo X inactivation to be able to dilute the amount of genes that are expressed in the X chromosome to be able to equal the expression between males and females. But there is a certain amount of genes that can escape X inactivation, leading to higher expression in females than in males. And perhaps this difference in expression of X-linked genes could actually perhaps explain why you could mechanistically have differences in NK cell homeostasis and effector function. So we looked at this database, DICE, and we looked at the expression in a large cohort of males and females in humans. And we found that there was a certain list of genes that actually showed sexual dimorphism, so higher expression in females than in males. And we wanted to confirm this with qPCR. And we looked at a lot of genes that seem to reproducibly be down in males compared to females in mice and humans. But we really were interested in this gene, KDM6A or UTX, because it showed the biggest difference between males and females here by qPCR and also in humans. When we stain for UTX with intracellular staining, we can see that females have higher expression levels by protein than males, which suggests that UTX levels are likely sexually dimorphic, and the increased expression seen in females is likely due to this escape from X-linked inactivation. And you can see that quantified here, that females have higher expression of UTX. So what is UTX and why do we care so much about it? Well, UTX is actually a histone demethylase. And so it can take regions of closed chromatin here that are marked by H3K27 trimethylated regions. It can cruise in and see these trimethylated regions, remove a methyl group, and then allow for accessible chromatin, thereby allowing for key transcription factors to bind these regions and then activate gene expression. So it's a really interesting idea that an epigenetic regulator is sexually dimorphic, and perhaps a altered epigenetic state could explain the differences between male and female NK cells. And one way that we wanted to confirm this is by making a mouse that was deficient for UTX using an NK cell specific CRE. We're calling these mice UTX-NKD mice. And you can see that intracellular staining, we reduce the amount of UTX. Although it's not all the way down to background levels, we think the antibody is a little sticky. But you can see that it's dramatically reduced. And we do this in female NK cells because there's also another gene called UTY that's expressed in males. And so we want to not necessarily study UTY function, we only want to study UTX function. This also has the added benefit that we can study UTX function in the absence of sex hormones, because now it's controlled for the same female sex hormones. So we can study UTX function. We can see that we have a dramatic reduction of UTX in these mice. And we know because it's in histone demethylase that there's likely going to be epigenetic differences. And that's exactly what we see. So if we look at combined ATAC-seq and RNA-seq, we see 6 main clusters of differential accessibility that seem to also translate into increased gene expression. And these genes were associated with effector function. So it seemed that the UTX deficient NK cells had decreased accessibility and expression of interferon gamma and other transcripts that seem to be associated with terminal maturation and effector function. Whereas they also had increased accessibility and expression of BCL2, which I've shown previously is really important for the homeostasis of NK cells. And so this started to make sense to us. Perhaps these UTX deficient NK cells might show similar trends and their differences in homeostasis and effector function. If we look more broadly at the transcription factor binding motifs that are now the differences in differential accessibility that are up in UTX, we can see enrichment for binding motifs in KLF family proteins, but a significant enrichment in the decreased accessibility for RUNX proteins, which Joseon has been shown really important for the homeostasis of NK cells. We also see a decreased amount of binding motifs that in regions that are more accessible in the UTX deficient NK cells for eomas and TBET, which are really important for NK cell homeostasis, but also effector function. So we're seeing a lot of differences in the epigenetic state, and also transcription factor binding that might necessitate differences in homeostasis and effector function, which may mirror the differences that we see between genders. And so we wanted to see if this is true in UTX deficient NK cells. And this is the only phenotype that I've ever seen in a knockout mouse where you actually have more NK cells. So in females that their NK cells have lower levels of UTX, actually have higher amounts of NK cells in the spleen. This is not just in the spleen as well. We looked in other peripheral organs. You have enhanced numbers in the spleen, blood, lungs, liver, and bone marrow, which was really suggested to us that these UTX deficient NK cells actually have enhanced cellular fitness. It's not just differential trafficking or accumulation in a certain organ. And this was corroborated by higher levels of BCL2, but also higher levels of BIM, but the BCL2 to BIM ratio was much higher in UTX deficient NK cells. So this suggests that UTX is actually restricting the fitness of NK cells through regulating BCL2 levels. And so we've shown that can happen directly at the epigenetic and transcriptional level. But I also mentioned previously that we see key differences at loci that are important for NK cell effector function. And by ATAC-seq, we can see that these UTX deficient NK cells have decreased accessibility at the interferon gamma locus, which also translates into lower amounts of interferon gamma transcript. We also see the similar thing at CSF2, also known as GM-CSF, which is really important in potentiating conventional type 1 dendritic cell responses to virus. We see decreased accessibility and almost no transcripts here of CSF2. So this suggests that UTX is actually epigenetically priming NK cell effector responses. So do we see that? If you stimulate NK cells with IL-12 and IL-18 ex vivo, you see a certain amount of production of interferon gamma, but NK cells that lack UTX produce less interferon gamma. And we can quantify this here. You get less interferon gamma positive cells, and they also produce less interferon gamma on a per cell basis, which suggests that UTX is epigenetically priming these effector loci in order to increase their effector function. And because we think that females have higher levels of UTX, this could explain why female NK cells show enhanced effector function. Everything that I've shown you so far is just stimulating NK cells with pro-inflammatory cytokines. You know, where's the in vivo evidence? Why do I care, right? And can this explain some differences that we'd see in susceptibility to viral infection? So we did that. We took wild type mice and also UTX deficient NK cell mice, and we infected them with a sublethal dose of MCMV. And you can see here that the wild type mice, they can control the infection, they can live no problem. But we could see that the mice that had NK cells deficient in UTX succumbed to MCMV infection at a sublethal dose, suggesting that their effector responses were suboptimal. And sure enough, if we use mixed bone marrow chimeras with wild type and UTX and NKD bone marrow, you can see that within each mouse, you have a lower production of interferon gamma of splenic Li4NH positive NK cells. And they also produce less interferon gamma on a per cell basis, which is consistent with data that I've shown you before with pro-inflammatory cytokines. Now, you could say at this point that, okay, well, UTX is a pleiotropic epigenetic regulator. You delete it, you're going to do something to NK cell development, and therefore these NK cells are screwy, and this doesn't really mean that UTX is controlling effector loci independent of its role during development. So we wanted to look at that, and we made a mouse model of inducible UTX deletion by crossing a ROSA26 ERT2 Cree mouse to our UTX flox mice, such that upon the addition of tamoxifen, you can now reduce UTX levels. And we made a mixed bone marrow chimera in order to be able to infect these mice such that they could both see the same level of virus, but we can interrogate the amount of interferon gamma they make in response to MCMV. So we made these mixed chimeras, and then we hit these mice with tamoxifen for 3 days before infecting with MCMV, and then harvesting NK cells 1.5 days later, excuse me, after MCMV. And what we found was pretty interesting. So the ROSA26 ERT2 Cree, it deletes, but it doesn't delete 100%. And so what we found was sort of a titration of UTX levels in each of these mice. But we saw a really interesting phenotype. The higher the UTX level was in these inducible UTX deficient NK cells, the higher amount of interferon gamma we saw. And so this trend really nicely and has a really strong correlation between UTX levels and interferon gamma. And this is also similar to what we see between the genders. So females have higher levels of UTX and higher interferon gamma on a per cell basis. Males have lower and therefore lower interferon gamma production. So we think that the levels of UTX are important for increasing the accessibility of these effector loci, thereby increasing the amount of transcripts that you can have to get a functional response in response to viral infection. So to sort of summarize what I've shown you from this part, UTX is a X-linked epigenetic regulator that shows differences between genders. So it has sexually dimorphic expression. It seems to control NK cell homeostasis by regulating BCL2 levels, but it can also modulate the accessibility of transcription factor binding motifs like TBET, EOMES, and RUNX KLF family members. It seems to also increase the accessibility of effector gene loci, such as interferon gamma and CSF2. There are others that I didn't talk about today, but we'll be submitting this paper, so have a look out for that in the future. UTX is really important during inflammation because it can basically set up a sort of pre-primed state epigenetically where NK cells have enhanced responsiveness to cytokines by increasing the accessibility of the interferon gamma locus. And if we tie this back to differences in sex, we can see that males have lower UTX expression levels, but higher levels of NK cells. And this is likely due to the effect that UTX repressing BCL2 expression. Yeah, that's true. And they have lower levels of interferon gamma production because UTX is positively associated with increased accessibility of the interferon gamma locus. In females, which have higher levels of UTX, since it escapes X-linked inactivation, they have lower levels of NK cells due to decreased BCL2, but higher levels of interferon gamma production due to increased accessibility of the interferon gamma locus. So we're really excited about these results, and really looking forward to any feedback on this part of the talk since it's unpublished. So with that, I will thank the people that did the work. Luke Riggin, as I mentioned before, is a very talented graduate student who recently graduated and is now in industry. Joey Lee is an MD PhD student who's done a lot of stimulation experiments with human NK cells. Our technician Rana, who's also done a lot of this work with sexual dimorphism and NK cell function. From the Pellegrini lab, Fei-Ying Ma has helped us out a lot with bioinformatics analysis, so single cell, attack-seq, and RNA-seq analysis. And Mandy Chang in the Su lab has sort of driven this sexual dimorphism project in collaboration with our lab and has done a lot of exciting work there. David Nathanson, Elizabeth Hernandez helped us out with BH3 profiling. Of course, we would be nothing without our fantastic UCLA cores, and of course, couldn't do any experiments without our sources of funding. So the second part of this talk is a wonderful collaboration between Maureen Su's group and mine. And here we are hanging out together in our summer barbecue, having a lot of fun. So I will stop there, and I will take any questions that are approved in the chat.
Yeah, so I have a couple just as we wait to see if people, and to see if that function works. So one of them is just a pretty basic question, and the second one is more of like a conceptual one, which is more fun maybe. So if you were to, so the first one is, if you were to overexpress UTX in male NK cells, would you expect to mirror that, or is there something intrinsic to the female mouse's physiology that is dictating those responses?
Yeah. So if we had a transgenic UTX in male, I would expect that we would see similar differences. Most likely because we see that there's no difference when we control for sex hormones. We've also done sort of kind of like a crazy experiment where we've made a mixed gender bone marrow chimera to see in male recipients if those differences still hold true, even in the setting where now you might have epigenetic remodeling by male sex hormones. So we're getting at that. I think we have the data, but still a little too fresh off the press for me to comment on. And then the second part was,
So then the second question is, I'm really struck by your RMP depletion experiment of BCL2. You only had modest depletion, but pretty dramatic effect. And so I'm as you know, there are multiple redundant cell death systems, for apoptosis, extrinsic and intrinsic. And for whatever reason, NK cells seem to be one of the rare cells that is pretty dominated by this modality. I'm wondering I'm wondering why there isn't a seeming extrinsic apoptosis pathway that is also relevant, or what you're thinking about this is there a reason this evolved to be really specific for that? Yeah.
Yeah, so I mean, NK cells are really, really important in recruitment of conventional type 1 dendritic cells, which, as you know, basically, if you don't have CDC1s, you don't have cross-presentation, you don't really have an effective T cell response. And so I think there's safeguards that are built into the homeostasis of innate lymphocytes, which make them a little bit more susceptible to apoptosis, because if you had these cells living long term in a lot of cell numbers, you'd actually get a lot of immune pathology. So I think it's probably something about that, just given their sort of bridging role between the innate and adaptive arms of the immune system.
Yeah, okay. I don't know. I think you might have given a perfect talk. I'm not seeing any. Let's give him just a minute here, but otherwise, I think and a decent number of viewers. So clearly, you crushed that. So if there are no other questions, please feel free to leave any of your questions directly on the YouTube stream or on the Twitter as well. The link that was up there, and Tim will do his best to get back to you as you watch them. And anything else, Tim? No, that was great. Great job. Thanks everyone for turning out today. Take care. All right, stream ended.